Hello, we shall be discussing on the next topic, but before that, let's clarify on what is bacteriophage or phage. Bacteriophage or phage are viruses that attack and cause lysis or break of the bacterial cell. Now, this is the structure of the bacteriophage or phage. A typical bacteriophage or phage contain a protein head consisting of a protein coat and a DNA. So the head of the bacteriophage or phage contain a protein coat and a DNA enclosing inside, and this portion indicates the neck of the bacteriophage, and these are the tails. So the genes the DNA of the bacteriophage is being protected by a protein coat, and this DNA is can be transferred to some other infected bacteria through this neck portion. So this bacteriophage attack or infect the bacteria by attaching itself to the bacterial cells through this tail. So once they attach themselves to the bacterial cell through this tail, they inject their DNA through this neck into the inside of the bacteria. So now we shall be discussing on the Hershey-Chase experiment. Mr. Hershey and Chase in 1952 conducted an experiment on T2 phage or T2 bacteriophage. They made two cultures, one culture containing the T2 phage bacteria with radioactive sulfur, that is S35, and on the second culture they mixed the phage, T2 phage together with bacteria and a radioactive phosphorus, so P32. Now in the first culture that is containing a mixture of this bacteriophage bacteria and the radioactive sulfur. The radioactive sulfur, that is S35, that is sulfur 35, gets incorporated into the protein coat of the phage, and in the second culture it is found that this phosphorus is the important components of the DNA. Therefore this radioactive phosphorus gets incorporated inside or into the DNA of the T2 phages. Now they allow the bacteriophage to infect the bacterial cell in the culture. So these are all taking place inside the two culture. In one culture it is containing radioactive sulfur, and in the another culture it is containing radioactive phosphorus. So the phage containing radioactive sulfur in the protein coat is allowed to infect the bacterial cell, and in the second culture the phage containing radioactive phosphorus in its DNA is also allowed to infect the bacterial cell. Now these two bacteriophage, that is the bacteriophage one containing the S35 and the other containing the 32P or the phosphorus 32, they begin to infect the bacterial cell, and after about 20 minutes of infection it is found that the bacterial cell begin to break and release the daughter cell of these phages. Now when these bacteriophages come and get attached to the bacterial cell, they inject their DNA inside this bacterial cell. So this is the this is the DNA of the phage, this is the DNA of the phage, and this is the DNA of the bacteria, this is the DNA of the bacteria. So in this culture the radioactive phosphorus 35 is only in the protein coat, hence there is no radioactive detected in the DNA of the phage. But in this culture the radioactive phosphorus is being incorporated into the DNA of the phage. Therefore the injected DNA of the phage will contain radioactive P32 or the phosphorus 32. Now the two these two phages after infection they get separated, and the first culture will contain the bacterial cells with no radioactive DNA. In the second medium, the culture will contain the bacterial cell together with the radioactive P2 in the DNA. Now, after some time, they isolated the phages and found that in the first culture, the phages contain the radioactive sulfur in its protein code, but in the second culture they found that no radioactivity is found because the radioactive phosphorus which is present in the DNA is being released inside the bacterial cell. So from this experiment it is being concluded that the DNA is the hereditary material because this DNA is being released into the bacterial cell in the second culture. So from this experiment it is found that the DNA is the hereditary genetic material.